Hello, everybody, and welcome to the second Shoal webinar of 2026. My name is Michael Edmonstone, and I'm the Communications and Engagement Lead here at Shoal. Shoal mobilizes urgent conservation attention for the world's most threatened freshwater species, and through the landmark 1000 Fishes initiative, aims to catalyze conservation attention for 1000 of the planet's most threatened freshwater fish species by 2035. Each year on the 3rd of March, World Wildlife Day, along with our partners IUCN Species Survival Commission, Freshwater Fish Specialist Group, and the California Academy of Sciences, Shoal releases a new species report highlighting the freshwater fishes described to science throughout the previous year. 2025 was a bumper crop with 309 freshwater fishes described, which is the most since 2017 and the third highest number since records began way back in 1758. If you've not had the chance to read it yet, I urge you to go and take a look when you can. There are some incredible species there and some really fantastic stories of exploration and taxonomic graft. Today, we're lucky to be joined by three experts who work extremely hard, often in very remote and sometimes really quite inhospitable places, to discover and describe some of these new species. First up, we'll speak with Bella Nagy. Bella is an independent researcher in biology, an explorer and storyteller with a deep passion for the African continent. Much of his work focuses on the seasonal killifish genus Nothabranchius, to which he added 5 new species in 2025, 4 of which were highlighted in the new species report. And fascinated by the legacy of early explorers and the mysteries of seasonal freshwater ecosystems, he has dedicated his research to the rare and elusive fishes inhabiting Africa's savanna pools. He also contributes regularly to conservation work as a collaborator with the IUCN, preparing red list assessments and co-authoring conservation-related publications. We will then hear from Dr. Nurjirana, or Jiran. Jiran is a postdoc researcher at the Museum Zoologicum Bogoriense, I hope I pronounced that correctly, MZB, the Research Center for Biosystematics and Evolution, BRIN, in Indonesia. She is a research associate at the Thematic Research Group Biogeography and Taxonomy of Endemic Fishes Research Group and the Aquatic Invasive Alien Species Research Group at Hasanuddin University in Indonesia. She is the lead researcher of the team that described Rennie's Styphodon. And finally, we will hear from Luiz Fernando Caserta Tencatt, a professor at the Universidade Federal de Mato Grosso do Sul. Luiz has devoted his scientific efforts to the understanding of the taxonomy of Callichthyidae and Loricariidae families of catfishes, especially the Corydoradinae and Hypostomus genera. Luiz was the lead researcher on the description of the ancestor Cory Hoplosoma noxium, which was the cover star of the New Species 2025 report. A very small bit of housekeeping. Today's webinar will last approximately an hour and a half, maybe up to 2 hours, depending on how many questions there are. And then after Bella, Jiran, and Luiz present, we'll have the chance for a Q&A session at the end. Please feel free. I see some people are writing on the chat at the moment, but please feel free to introduce yourself and say where you're dialing in from. It's always exciting to know which far-flung places you're watching from. And keep your questions coming in throughout the talks. The session is being recorded and we'll upload it to Shoal's YouTube channel and website. Bella, Jiran, Luiz, it's fantastic to have you on board. Ruth, I have just seen, has raised her hand. If we can, we'll keep the questions to the end, but if you do have a question, make a note of it, and then at the end, when everyone's finished, we will move on to the questions. But great to see you here, Ruth. Cool. Bella, if you're ready to share your screen, it's over to you.
Yes, thank you very much. Do you hear me, Michael?
Yes, perfect. Thank you.
Okay, do you see my screen as well?
Yes.
Okay, perfect. So thank you very much for the introduction, Michael, and happy to see you all, guys. I see that there is a very international audience. Actually, I'm Hungarian, but living in France. So, hello to everyone from the Paris area. I would like to tell you a couple of stories about some of those new species that have been described last year, but not only. We discussed with Michael when we prepared the publication that there are so many interesting stories, finding new fish or rediscovering old fish that nobody has seen since very, very long time. So the idea was that I show you just a couple of those stories that how we get those fishes known or even into the aquariums. So basically, I'm talking about notobranchous fishes. Those are seasonal fishes. And here just a few species. There are about 100 of them. Very, very colorful, small, like your finger. And they are living in Africa. So where you see here the squares, that is where those fishes are distributed, in eastern, northeastern, and southeastern Africa. Here the color, the darker the color, it shows that more and more species are living in that particular area. And it is not only that there are in Africa those species, but unfortunately, they are getting more and more in danger. So you see that there are about 100 species. So when you see the percentages, those are approximately number of species also. So 4 of them are critically endangered, 23% endangered, and so on and so on. So over 70% are in the threatened category. And that is obviously a problem. Those seasonal fishes, they are living in relatively small areas in many, many cases. And that is one of the reasons that they are in those categories. As I said, they are seasonal. So, in most part of Africa, there is one rainy season and one long dry season per year in those areas where there are 2 seasons. And that means that this circle, what you see here, it represents 1 year of life or life cycle of the fish. It starts above on the top, where the rains are coming and the fish is growing very quickly, and then spawning, putting their own eggs into the substrate, and then the egg is surviving in the substrate to hatch again when the rain is coming. And that is an important aspect because, if anyone wants to see those fishes, need to go there when they are alive in the biotop. And I have been 3 times in Congo, and I've been in many other countries of Africa as well. And I'd like to show you just one short story from each of those trips, which is linked to the rediscovery or discovery of a species. So you see here a painting, actually, where women are collecting with a basket some fish, fishing there. And it happens like this also in real life. Here you see that they are using the basket, and then when you see the basket, there are some fish inside. So if you see those colorful males, then those are notobranchous fishes, which with the help of these local ladies, we have been able to collect. By the way, it was a new species as well, but this is not the story. In Congo, most of the notobranchous fish are living in the southeastern part of the country, which is the former Katanga province. And there was one fish there which was very, well known by name, but nobody has seen it since very long time, Notobranchus brieni. It was found in 1937, described in 1938, so almost 100 years from now. But at the time of my first visit in that country, 2013, it was 75 years old. The museum specimens had no color anymore. The only thing what we knew is that they are coming from Bukama. So with the help of the local university colleagues over there, I was flying to Lubumbashi, and then from Lubumbashi on the road to Bukama. So this is 600 kilometers. And, it might sound easy if you live, I don't know, in Germany to take the Autobahn for 600 kilometers, which is just a few hours. But, if you go to remote areas and also looking for fish which are in temporary waters, then everything is underwater, obviously, and the road conditions are different. So we thought that if we are using 2 full days, from dark until dark, then we could get there during 2 days. And then this is what happened, that we started out. And at the beginning, I was happily taking photos and even videos, about the conditions. But then later on, I needed to pack away my camera because all the water came into the car. and then, blocking trucks blocking the road, so we need to drive into the grass or into the trees actually, and going through. But this is what we did, for 2 days. Then we arrived at the Lualaba River, which is part of the Upper Congo, and then we used the ferry to go to the other side, and we were not very far anymore. Out of the 600 kilometers, there were 45 left after this ferry. And then when we got away from the ferry, only 45 kilometers, and then our car broke down after 2 days. It was late afternoon in this moment. And I don't know, stub axle, I think it's called in English, which was broken. So no more progress on that day. So, some of us, took a nap for the night in the car or in the stand before the car. And then it is me, my legs, and one of my colleagues, Emmanuel, taking the night. And early morning, because we could make a call, and early morning, a fisher person came from Bukama, 45 kilometers, by a motorcycle. So this is early morning next day. I mean, you see on my face that it was not my best night ever. And, I took my backpack and some net, and then this very kind person who came all the night, so he started at midnight to get here, 5 in the morning when the first sunshine is up. And then, I put myself on his motorbike and going to Bukama, 45 kilometers. It was a, adventure when you think about it, taking those very bad roads and then just to do the left, the remaining 45 kilometers. On the other hand, after a few kilometers, the guy told me that he knows a shortcut. And then, first I thought it's a good idea, but actually I didn't know that what that means is that the shortcut means that you take a known but very bad road to an unknown and even worse road. And this is what we did. So guys, when I'm talking to you about this day, what happened, my back is still hurting in that moment, when I'm just talking about it. In Bukama, I already have been booked in the best hotel in the town, which is the Hotel Deluxe. And I got the biggest room in that hotel, which was perfect, but I can tell you I didn't stay a minute because went out right away. And here this is a satellite image of Bukama itself. And in the middle of the picture, just beside the Congo River, you see a bridge crossing the Congo. That is a railway bridge, so only for trains. And on the south side there is a train station. And then you see in the middle of the village that there is a swamp. And then I thought that is a very good place to start with, because this is the type locality of that fish that nobody has seen for 75 or 80 years. And actually, this is the swamp. You see in the background the train station, the railway station, the electronic infrastructure. And then here, we found, or I found, because I was alone with my motorbike, the Notobranchius brieni, which was never seen before in terms of colors, or never photographed. And this is the short story of a rediscovery of that. Since that moment, fortunately, the fish is still in the hobby, and there are interested people who are breeding it. I went back to Congo in 2016, and I would like to show you a different trip. So from Lubumbashi again, I wanted to go with the colleagues, professors from the university in Lubumbashi to the area around Lake Mueru. And why it was important? Because this area is called the Triangle de la Mort. So it is the triangle of death in the local language, but because of different instabilities which might happen in that area. But that also means that it was not well researched beforehand. So it's always a good chance to go there, and accompanying local people, it also gives the security, that we can ask around, and if anything happens, then, we can make it safely. But the road conditions are, a little bit comparable. Not the first 100 kilometers. So the first 100 kilometers, so we took all the what necessary stuff from the university, and then on the road, first 100 kilometers is like this perfect road. If you see something, but then after 100 kilometers, we went on the dirt road, which is for the rest of the trip. But there is a kind of protection, a warning that you see on the picture. This is a rain barrier. And just, beside the very big tree on the picture, you see the barrier that it is up, so it is opened. It means everything is fine. You can cross the area, and this is a safe area to travel. So we continued, and probably 5 minutes later, we find this. This is a bridge. Obviously, we will need still to go to several hundreds of kilometers, so we will need to cross it. So I took my boots, and then we tried to, investigate if it is stable enough or not, and actually it was. And then that means that we could make, we took one vehicle of the university, and then we could cross the bridge. So actually it was good. And then, we arrived to places like this. It is not, I show it only once, but it was several of them, that when the road is bad, meaning that there is mud and all the vehicles are stuck in the mud, then there is a construction work which needs to be done. And here you see that it was a Chinese contract to take care about this road. So you see the engineer there and everybody working. And what do they do in that moment is that they make a big hole. And then when the hole is big, then they take out the water from this hole. And then they bring some dry substratum from somewhere else, and they put it into the hole. So then it is repaired. I mean, you can imagine it takes like, 3-4 days to do that. And here we are in the middle of that. When we reach that point, there are already buses and then trucks waiting since 2 days at this point, and nobody can cross. So we were lucky in this moment because, local people told us that, they can help us. And please, conservation people, do not look very carefully, but here we needed to cross, unfortunately, a local forest. On the other hand, that was the only possibility to proceed into those regions where, we could do our investigation. And actually, our car was the smallest. The other cars here, they have been trucks and buses. So we were the only car who could cross this small forest, which was around the big hole. And we had that kind of situation several times. Anyway, we crossed it, and then, what happens is that you get a flat tire, but that is probably 20 minutes just to change the tire. So nothing compared to the 3-4 days, but you need to wait, together. So we get to the area of the Lake Mueru, into the triangle area, and then we start our investigation. And here we see that, you see that is a kind of temporary pond in a river channel. And the water is almost staying, not moving so much anymore. And there are some like channels or roads built from the vegetation. So we asked the people if there are fish traps, and they said yes. So here one local fisher is takes out the trap, looking into it, like here. We just asked if there are some small fish inside or not, just to have an idea what is living in this area. And then they said yes. So, we look into that, what's going on. This is a very good solution to get a quick idea about, what is living in that area. And then other fishers, they have seen that we are interested. They have brought more and more fish. And then, I was looking through many of them. And first, I didn't find anything. Here you see me in action. But then, there was one fish which was interesting, I think, and that was this fish. And actually, this is a female. So you see that compared to the colorful males, this female does not have, specific color. So this is a Notobranchius. Certainly, but we didn't know which species, and we are in an area where nobody has been before for this kind of fish. So that was worse enough for us to get into the water and then to try to identify what's living there. And where is a male, where you can see the coloration. So there is Professor August Chachamanda from University of Lubumbashi, and another person from Europe, Gabor. He was a collecting mate on a couple of trips with me, and we went into the water to see what's going on. And actually we found a fish, and that was a new species for Notobranchius. So it was very successful. And it was worth, going through all those adventures. We have seen, when after this, I go around the habitat and then try to do some photographs also from different angles. I see that there are some males, actually, I didn't see them before, hidden, in the grass, and they are just thrown away. And it turned out that all the other fish, what you have seen also in the photos and the video, the local people eat them, but not this one, because this is too colorful and they think it is poisonous. So, bright colors, they are always a sign of some danger. And by the way, the local name of this fish is kikende, which is the name of this clothes, what the women wear, because they are also colorful, like the male of the Notobranchius, and they use the local name for that. And the last story, in 2023, I went back again to Congo. And first, I wanted to show you something which is very interesting from that area. You know that Congo is important also for locally, but also for Western interest, because of all the minerals which are found in that country. And what is specific to that area is that all those minerals, they are on the top, on the surface. So you do not need to drill down for that. You can basically very easily take it away. You can see them. I mean, you can see that also by color. I think on the left side, that bluish is copper, and then on the right side, that is probably cobalt. I think I'm not an expert of that, but my local colleagues, they are always telling me by visual. By the colors, what kind of minerals you see there. And that is also a problem, because many of those minerals are getting into the water. And local scientists, they made investigation and publication about it, that those minerals, they are accumulating in muscles of the fish, for example, and that is bad for many things. Obviously, it is deteriorating their immune system, but also people, small fishes, they also get, a non-healthy portion of those minerals. So anyway, but I would like to show you what can be caused by those. So again, one trip, going back to the triangle, a few years later, it was still very complicated. A lot of cars, small buses, trucks blocking the road. So we had no other choice but then take them away from the main lane of the road. I'm trying to. It was not the first attempt, but finally, we succeeded to do that. And once the bus is away from the lane, then you see that the bus is already, the small bus is just behind, and then obviously we can take the same lane, the same spot, and cross it with our car easily. So that is what we needed to do again a lot of times. Here another example where a small truck is blocking our way, so we need to have them out, getting away, and then cross it at that place, which is then made. Just to show you, I mean, there have been many occasions like this. So again, a couple of days of travel to get to the area which is interesting for us in terms of the research. Bad road everywhere. This is not a surprise, just at the end of the rainy season, this is when we need to go to find the fish. So we have no other choice. We cannot go into dry season. Here, I don't know anymore how we got through. And we found our way somehow, just a few pictures. I have much, much more than that. But so we arrived to an area. This is a picture from last time, so 2016, where we found a fish, and it seems to be a new species. And the problem was, here we are taking some photos of the fish, and the problem was that the fish was sick. And this is what that contamination, the minerals can cause, that the immune system is not the same anymore, and they can easily be sick. This is a phenomenon which I only see in Congo and not in the other countries of Africa where those seasonal fishes are living. And I draw it back, to the contamination, which is particularly strong in that area. So in 2023, after we have been drawn out pulled out from the mud so many cars, we arrived at the same place. And here we found the fish again, and this time it was healthy. So we could make good examination with live fish. And it is not only morphometrics, but also chromosomes and many other phylogenetic examinations. And this is the fish already at home, photographed in my tank. And that is one of those new species, which you have seen also in the new species publication of shoal that have been described last year. And the pathogen which we found, it was actually an Aphanomyces species. I made the genetic identification, and Aphanomyces invadans and laevis, two well-known pathogens, now also in Africa. They are spread around the world, and those populations which I found, those are marked with the red numbers. Those are my field codes which I used in Congo. And if anyone is interested, I made a publication about that as well, first time on notobranchius fishes identified. And last but not least, this is one of the publications where 5 species have been described. So thank you very much, Michael. You guys, you picked it up in your new species presentation or publication. And you have seen now, some of those short discoveries. Obviously, I can talk hours and days about all those adventures, but it was just a very short introduction for you. If anyone is interested to see more about notobranchius fishes, those seasonal fishes, then visit my website, Wild Notos, or buy, books. One of them is Rough Road into the Deep Unknown. The motorcycle adventure is written in there, and many other adventures, when I was arrested in some countries and so on and so on. So that is already available. And on the right side, Return to the Land of Rains, that is coming out in 10 days. So anyone interested in those adventures, please don't hesitate to look into those books. Thank you very much, guys, for your interest. And then, if you have any questions, just put it into the chat or give it to Michael, and then I will stay a bit to try and answering them. Thank you, guys.
Brilliant. Thank you so much, Bella. What a colourful insight into how challenging it can be on these expeditions. And I guess an insight into how many species are likely to still be undiscovered and undescribed in these sort of very remote regions of the world. I know when we were messaging before writing the new species report, you said that one of the main reasons that you sort of went to that area is this road has opened up the area. And as there's more infrastructure and more development, it obviously allows the areas to be opened up much more easily and allows the species description. But on the flip side of that coin, the development leads to anthropogenic pressures on the habitats. So yeah, as soon as some of these species get described, the very act of sort of being able to describe them is partly the thing that is can be causing threats as well. Yeah, thank you for that talk. Jiran, are you ready to share your screen?
Yeah, sure. Can I share screen, Ram?
Great.
It's already show.
Yeah, perfect.
Okay. Hi, everyone. Good evening from Indonesia. Thanks, Michael, from Seoul for organizing these meaningful webinars. And it's a pleasure to be here and share this fascination journey with all of you. And today I will take you through the story of small yet remarkable freshwater goby known in the aquarium world long before it gained scientific recognition. This journey of Rhinistipodon, a species that bridges between hobbies discover and formal taxonomy. Through this talk, we will explore how to curiosity, collaboration, and scientific come together to reveal the biodiversity that was hiding in plain sight. And let me introduce myself. My name is Nurjirana. You can call me as Jiran, and I am honored to share this story with you. Yeah, for my introduction, freshwater ornamental fish from Asia are highly popular, particularly goby, which has know their small size and striking coloration. These fish are widely tried and commonly used to enhance aquascape collection among hobbies, and many species are promoted through social media platforms such as Instagram and Facebook to attract buyers. As a researcher focusing on Amphidromus gobies, I spend a lot of time not only in the file or museum collection, but also talking to the office collector, or in Indonesian we call it hunter, and aquarium hobbies. This collaboration allow knowledge exchange between science and the aquarium trade, and because sometimes they discover things and before we go. And yeah, ornamental fish trade in Asia is very popular. Today I will focus to share Arenistipodan story. Is a widely species popular in aquarium market across Southeast Asia. And before being scientifically described, this fish was exported from Sumatra, Indonesia, under the trade name Stipodon spp, and commonly known among hobbies as pink Stipodon. And it was sold at relatively high price, ranging from 10 to 17 US dollar per individual. From Indonesian course is very pricey. So with export volumes of 100 to 510 individual per shipment, making it economically attractive for collectors. And yeah, this image were obtained from collector and hobbies and widely circulate on social media. Many of them consult with me regarding species identification. In return, I gather valuable data on species occurrence and collection localities, which helps track distribution pattern in Indonesia. And they creates a mutual beneficial relationship. And interestingly, many goby species may already be traded before being formally described. So, early discovery for this species, despite its popularity in aquarium trade, this species was first collected during the Enggano Island expedition in 2015, conducted in National Research and Innovation Agency, or in Indonesia we call as BRIN. At the time, it was recorded only as Stipodon sp in this report, and along with several other unidentified species, recognized but unnamed, and discovered but not yet understood. So, let Rainy Kurniadiati for his article on a local article is Ichthyologist fish from Enggano Island. Report this fish candidate new species but remains undescribed. Following her passing in 2019, the species remained unresolved. So in 2022, during my postdoctoral research at MZB, I rediscovered the preserved specimen, and then given my focus on Amphidromus goby, I initiated the formal description of this species and collaborated with colleagues from MZB. Then the data from Reni Kurnia Hadiati before, this study was based on a previous work by Hadiati and Sauri 2017, including specimen data, habitat, and locality information. to describe a species, we don't rely on appearance, but we measure, we compare, and using morphometric and the meristic analysis, we combine standard morphometric measurement and the meristic counts, following established taxonomy method and like a type of locality and some data of from this expedition. And yeah, previously I focused study for species Amphidoros gomi. There are 5 genera in Indonesia, and like is Deadly-Scapus, Middle-Scapus, Lentipes, and Sicyopterus, and the last Stipodon with some characteristic. We can Stipodon we can see in from the teeth and for the skeleton is specifically for goby. And yeah, there are some Stipodon widely distribution across to the Indo-Pacific regions. Indonesian is one of global center of diversity for Amphidoros goby with several endemic species. Then private study classifies Stipodon into 2 major species group based on morphological and coloration pattern. Cat et al. 2011 analysis 6 species Stipodon and divide them to clades. There are Stipodon elegans species group with 14 to 16 pectoral fin rays and large body size, generally 4 to 7 centimeter only for standard length, and Stipodon subprenus group with mainly 13 to 14 pectoral fin rays and smaller body size, generally less than 4 centimeter for the 2 species group. Yeah, Stipodon from Enggano Island, Sumatra, Indonesia. The description of Stipodon hadiati based on preserved specimen we conduct, and we did morphological examination, radiography, and analysis of sensory papilla pattern. This method validate to diagnosis character. Then yeah, this is my morphometric and meristic character of Stipodon from Enggano Island. We do there with this strong character in the from the fourth spine and first dorsal fin is filamentous and may extend to the second dorsal. This a strong character for this species we found. Then yeah, there are similar species with this species I found. Yeah, therefore spine. This trait is similar to found outside Indonesia, such as Stipodon saperinus from Vanuatu, New Caledonia, and Fiji. And the second one is Stipodon tuivi for Marquesas Island, French Polynesia. Is there are similar character for this first dorsal longer than species found from Indonesia. Yeah, we compare meristic character and for Stipodon from Indonesia and from Stipodon from the world, and all characters is significantly different for other. And with the highlight data, the red triangle that shows Stipodon hadiati differs significantly from other Indonesian and global species. For our publication with selection of meristic character, we provide premaxillary teeth. It's a different. There are different range for other species and number of scale in transfer forward series also, and number of scale in transfer backward and number of zigzag series also. So yeah, this is our provide on my publication also. We compare morphometric relationship for species from Indonesia, including caudal fin length, second dorsal fin length, and anal fin length, and Stipodon hadiati with right side heel. It's significantly different for other species. Yeah, there are, as 2025, there are 24 recognized species Stipodon worldwide, and Indonesia has a significant portion of this diversity, including several endemic. We also provide update information. I highlight the blue here. This species report from Indonesia. We also provide update information as synonym, distribution, common names, and conservation status. And this Stipodon Hadati become a part of global record and named the 100 scientists who was made this discovery possible. We dedicate this species from late Reni Kurnia Hadiati as a Indonesian taxonomist and one of my role model taxonomists from Indonesia also. Yeah, there are 12 species Stipodon from Indonesia in 2025. Many of which are highly attractive and popular in the aquarium trade. Stipodon hadiati represent the 34th species described globally. Indonesia is home of too many stunning Stipodon species. Their color attract hobbies worldwide and popularity come with a cost. Unfortunately, yeah, we know that Stipodon, one of Amphidromus gobies species in the world, and so many species in the Indo-Pacific. Most gobies trade in Indonesia, including Stipodon, are Amphidromus species, migrating between freshwater and marine environments during their life cycle. Their survival depends on connectivity, on the ability to migrate. And in Indonesia, yeah, post-larva harvesting, but as post-larva in this species is target intensive for consumption. Not only Indonesia, but some area in the Pacific area, post-larva gobies are harvested intensively during upstream migration, and these are used for consumption and sold as a high-value food product, forming an important local tradition. Yeah, this is for food for trade. This a tradition, but also a growing pressure for this species. And for adult species, this Stipodon become target intensive fishing for ornamental fish. We know both of post-larva and adult gobies are heavily exploited for food and the aquarium trade. Also, we are describing species faster than we can protect them. However, most newly described species like study on ecology and biology, population dynamic, and habitat assessment, and conservation status. But the species we describe is just describe or report like distribution, not further research after that. So this created a challenge in evaluation to their vulnerability, especially under threats such as overfishing, habitat degradation, river damming without fishway. So many dam in Indonesia without fishway, and pollution, including climate change also. So the question is not longer just how many species exist, but how many will survive for this species. Yeah, the story of Stipodon, rainy Stipodon is a highly important reality. Biodiversity can be discovered not only in remote ecosystem, but also within the aquarium trade. And this journey demonstrates the importance of collaboration between scientists, hobbies, and local collection. Moving forward, integrating taxonomy, ecology, and conservation is essential to ensure the sustainability of Pedromus gobi, is especially in biodiversity-rich region like Indonesia. Because in the end, we are just naming species, we are telling their story before they disappear, and yeah, that's in Indonesian case is about of is high exploit intensive for fishing. Then yeah, this our team for this described species rainy Stipodon, and there is colleagues late Rainy Kurnia Hadiati at MZB during my postdoc research at MZB. And I think that's all for my presentation. Thank you for your attention, and I look forward to discussion and collaboration. think that's it. Thanks, Mike.
Thank you so much, Joanne. Sorry to interrupt you there at the end. They're such colorful and charismatic little fishes. I can totally understand why they're so popular in the aquarium hobby. And I know when we come onto the Q&A at the end, there's a bit of discussion about the aquarium hobby's role in conservation.
Yes.
Luis, are you ready to share your screen?
Yes, sure. Can you see the presentation?
Yeah. It'd be great if you could go to presenter mode, but yeah, I can see it. We can see it.
Is it okay now?
We can, yeah, that's it. Perfect. Thank you.
Okay. That's it. Thank you. Can I start?
Yep, all good to go.
Yeah, thank you. First of all, I want to thank you, Michael and Shoal, for having interest in my work and for choosing Oplosomonoxium for the cover of this year's show. It was a very nice surprise. So you're here to talk about a little bit of the story of how we managed to capture Oplosomonoxium, and a little bit about the area itself, which is a very interesting and nice place in Brazilian Amazon. So who are the Callichthyidae catfishes? They are very, very interesting fish from the Neotropics, and you can easily recognize them among the other groups of catfishes because they have two series of longitudinal dermal plates. So they're the only group presenting this feature. So you cannot misidentify them. And here is the actual look of this armor, you know. So you have the superior row and then the inferior row. And they are very hard, but also some elasticity. So they're very, very resistant to impacts and bites and everything. So it's a pretty good armor. And it covers almost all of the fish body. And now we have about 230 species. And then we have two main groups among them, the Callichthyidae, which are a bit larger. They are very appreciated in Brazil as a food and also as a live bait for big catfishes. So in some regions of Brazil, you find them in they do kind of soup with them. They are not that big, you know. They can reach about, I think, 15 centimeters, maybe 20 centimeters, but they are very appreciated economically in Brazil. In the aquarium hobby, you can find them, but not as strong as the Corydoradinae. And they are now with 17 valid species. It's a pretty smaller group. And then we have the Corydoradinae catfishes, which is the target of our meeting today. These are much smaller group, and they are very species-rich group with more than 200 species right now, and they're very appreciated in their aquarium hobby. As you can see, the colors are much more vibrant than the average Callichthyidae pattern. So here's two main groups working with them. I started with Ann Nyssen and Isaac Spruker, and they worked for about 30 years. And now we have our group since 98. with Marcelo Brito. So we have in history, these are the two main groups of people working with this group. And how exactly we find them? The first option is you go to museums or collections so you can check material, and sometimes eventually you find new species hidden in old collections. But considering how appreciated they are in the hobby, we are always receiving new images of fish being exported, and the flow is much bigger than we can see in museums or collections. So the best thing to do is you look at the map and you look at this red area. It's just an example. So you have a well-sampled area and this patch, this blue patch, non-explored. So you go to these non-explored places to find them. So most of them are in very hard places to reach for different reasons. Sometimes because drive or flight to these places is difficult, or because some local situations are complicated. So most of the undescribed fishes are not in easy places to go. You have to reach quite difficult places to find them. The Rio Tapajós is one of the biggest tributaries of the Rio Amazonas. And it's a wonderful place if you like Corydoradinae, because I think it's the most diverse place for the group. So here are some examples of fish known in the hobby from there. Some of those are described already. Most of them are not described yet. So in the hobby, we have this code system, which will recognize the species mostly by color pattern, geography, locality means. So we have a lot of coded species from the Tapajós. And then we started a project about, I think, 6 or 7 years now. And we had 2 main expeditions to collect Corydoradinae catfishes in the Tapajós. Both of them were mostly financed by aquarium groups. So here you can see 3 main groups that were very important for these expeditions to happen. So, and some of the UK and 2 in the USA. So it's not exactly a hard trip, but it's a bit tiring because distance. So I was housed by the time in Cuiabá, which is in central Brazil, and is Red Star. And then we flew to Santarém in Pará, and then we took a car to Jacarecanga region. You cannot fly directly to Jacarecanga because it's a very small city in a remote place. So you can go to Santarém or Itaituba, 2 big cities in Pará, and from there you can pick a car and go from the Transamazônica, which is a pretty hard road, similar to those that Bella showed us earlier in rainy season. Thank God we can travel in the dry season to capture the Corydoradinae. So the road is not exactly that bad in the dry season, but it's a 2,000 kilometers travel. So Jacarecanga is a very small city. You can see its location in the red dot in North Brazil. It's as you can see, the number of inhabitants. It's very, very small, for at least for the patterns in Brazil. It's a region very important for ecotourism, sport fishing. It's a nice region if you like the indigenous culture, if you want to learn more about the indigenous people from the Amazon. It's a nice place. And it's also a very the activities of gold mining is very strong in the region. So people mine for gold there, which is mostly the main impact right now for the biodiversity of the local fauna. So here we have some sampling sites. As you can see, this first place is you can always see some signs of human life. People they like to dive in these places and swim and everything. It's a place for resting after a working day, fishing, and is also the place where we captured most of our specimens. Here you can see some of the places are basically inside the city. Thanks to the, I think, because it's not a modern city with industries and everything, the impact from the people, from the locals, are very low. But the gold mining activities is very strong in some places. So here you can see people taking a bath in the river. Here is another sampling site. This is literally inside the cities in the other side of this picture. You can see another one. This is a small place for fishing. Most of the specimens we capture from Oplizoma auction were from this exact site. Here is another one. This is a lake-like collecting site. And so what we got from there, we got a really good batch of cories. So 13 species from there. Some of them described, as you can see on the left, and on the right you can see some of the coded species, which are still under analysis or under description right now. And then you can see the 3 red names are species we described from our project. So you have Corydoras caramater and both Oplizoma species. Noxium and Tenebrosum were described in the same paper. So here you can see a picture of Corydoras caramater. This is quite common species in the region. We described this in 2024. And then Oplizoma noxium and Oplizoma tenebrosum, which were described also in 2024. So the star of days of Oplizoma noxium. Those are very interesting species by many reasons, mostly because of the color pattern of them is not exactly usual in the group, because of these pale blotches on the body mixed with the dark blotches. So this is pretty rare to find in the group. So for the locals, they call it the Corydora jabuti. Jabuti is this small guy on the left. I don't know why they consider them similar. They say they have general appearance like the jabuti. And for the hobby, you can see them called by the name ancestor cory because of this different color pattern they have, this peculiar color pattern they have. And well, people consider it some kind of a primitive or ancestral color pattern. So you can see these guys are very interesting, you know, not only Noxian, but all Corydoradinae in general. Now we have some exceptions, but in general, they are both venomous and poisonous. You can see here in the shoulder of the fish, pale area where the axillary gland is located. And for them, this gland is huge. It occupies most of the clatrum, which is rogally the shoulder of the fish. And here you can see in the axillary region, we have an opening, a big opening where the toxin is released. It's released in the water, and also it will cover the pectoral spine. So they have a quite painful sting. Here you can see the dorsal spine and the pectoral spine. They have serrations. They are quite pungent. So the toxin released in the axillary region will cover the spine, and if he stings you, you feel a lot of pain. It's a quite painful sting. The pain lasts about 5 to 10 minutes, and it's the place generally gets red, and you feel like a heart beating in the area. So it's a quite painful sting. When we talked to the local fishermen, they said there is no other Corydoradinae in the region with a similar sting. They said this is the most powerful sting in the region, and they also have documented a lot of problems of them killing other fishes. So the water they are in stress, they get foamy. As you can see, this is just an example, but you can see the water become milky and foamy, and they can kill other fishes very quick. So you have to, they are quite resistant to their own toxin. They eventually can kill themselves, but they're much more resistant than the other fishes. So you have to keep them only among the same species. If you mix them with anything else, will die first. So it's a species you have to be cautious when you are transporting them, and we always do the transport in isolating them from the other fishes. So their behavior is pretty unusual, different from most Corydoradinae catfishes. They like to live among leaves, branches. Usually you find the cories, as we say, in sandy bottoms, in clear water places, and they are not like this. And it's interesting because they are found among these leaves and also these branches, submerged branches. They don't swim a lot. They stay put on the ground, and also during the day you can do wherever you can in the places you won't find them. It's like they don't exist in the place in the morning. So when you go at night, you can see them. Interestingly, they don't form shoals. They live a little bit isolated from one another, and very with slow movements, and they swim a little bit and stop, and they don't. We always find them associating with the marginal part of the streams. So it's a quite unusual behavior for a Corydoradinae. They are quite active during the day generally. So you can see them, but they are very aggressive during the day. So if you want to capture them, you go at night, and it's easier to capture them. But in this case, this guy, you have to go at night and wait, and it's interesting because they live in difficult places to reach with nets, with small nets, hand nets. So the locals collect them with the hands. It's very interesting. So the Rio Tapajós project is far from being done. We have many more species to find and describe. Here are some examples of fish popping into the aquarium hobby. It's a fight. It's an unfair fight because we're always working, and then the hobby appears with new possible species. So we're trying to reach the expectations of the hobbies, but it's quite difficult because they discovered this new species much more consistently than us. And we have this guy still needs to be studied because it's a very I don't like to use the name, but it's a pretty important fish for the hobby because it's a very emblematic fish, the zebra Corydoras, which is an oplisoma right now, but we have to find it and potentially describe it. It seems indeed to be an undescribed species, very beautiful. And well, we have a lot of work to do. And just to end the presentation, I want to thank these guys, this hobbyist groups, for supporting my work in the past. It's about 15 years now of work, and they are always with me, supporting me in many ways. It's not only helping to fund expeditions, but some things which are we don't imagine sometimes, but we're talking about English language review of the papers. We have books, literature, information, pictures, everything. So I'm very grateful and honored to be considered by them as worth of receiving this attention. So thank you very much. And if you have any questions, I'll be happy to answer. Thank you, Michael, again for having me.
Brilliant. Thank you so much, Luis. There were a question and a point from Pete Liptrot. Great to see you here, Pete. I'm actually not sure which species he's referring to, but he says these are available in the aquarium trade as giving some CB young. I found them to be amongst the most shy Corydoradinae I've ever worked with, and can confirm their sting is effective. It sounds like Peter's talking from first-hand experience there. And he also asks, why GCF pygmaeus? Is there a cryptic diversity within this taxon?
Yes, I think he's talking about the Noxian in the last comment. It's indeed a very shy species. It's a very peculiar species in behavior, and I think the hobbies will have a bit of trouble to reproduce the places and everything they need to, because they are very different from the others. And about pygmaeus, it's because the locality itself is a bit far from the actual type locality of pygmaeus, but morphologically they are very, very similar, and I was not able to propose any potential difference. But considering the different locality, we are still looking. But I think it will end up being true pygmaeus, just a new record for the species.
Okay, brilliant. Thank you. And there were a couple of questions for Jiran as well. Dominique Sotti asks about the Renistyphodon. Excuse me. What is the environment of the fish? What kind of water? What kind of flow?
Yeah, for Stiphodon are specialists of clean. These are typically found in clear and fast-flowing freshwater stream, usually in tropical island environment like Indonesia. Of course, the habitats with the substrate like ribble or pebbles in the river, and the most found in the middle stream, lower and middle stream of the river for the Stiphodon. And the second question I answered also, right? Or
Yeah, I'll just read it out from the one from Stephen.
okay.
Yeah, so Stephen Grant, who I suppose is the same Stephen Grant that has supported Luis's work. So if so, thanks for joining the webinar, Stephen. Stephen asks, are the gobies being illegally collected for the aquarium trade?
Okay, that's very important question. In Indonesia, not all gobies in the aquarium trade are collected illegally. Many are harvested by local communities as part of small-scale fisheries. In some cases, this activity is not strictly regulated rather than explicitly illegal. However, the concern lies in the lack of clear management, monitoring, and traceability. For many Amphidromus goby, including Stiphodon, we often don't have defined quotas and specific regulation and proper documentation of collection site. For this creates a gray area where collection may be legal in practice, but not necessarily sustainable. And there are also cases where collection occurs in protected areas or involves species that have not yet been assessed, which raise conservation concern. Also, rather than framing it simply see as illegal or legal, I would say the bigger issue is whether the collection is sustainable and well managed. Moving forward, improving regulation, traceability, and collaboration with local collector will be essential to ensure to the aquarium trade does not negatively impact whale population. This is for me.
Okay, brilliant. Thank you so much. And that actually leads on to sort of a wider discussion about these species and these genera and the aquarium trade. Not through design, the three experts that we have on the panel have described species that are part of genera that are very popular in the aquarium trade. So Corydoras, gobies, killifish, extremely popular. And I'll ask this question to you, Luis, in the first instance, and then Giran and Bella, if you'd like to jump in at any point as well. What role do the aquarium hobby have to play in your research?
Well, for me, it changed everything, when I first contacted the hobbies for the first time. In academy, we have in the past a wrong view that we shouldn't work together with hobbies, this is a very wrong perception, because indeed in the past there were problems regarding some hobbyist scientists, and well, but this is far from the truth right now. They are first, they are very knowledgeable people regarding the fish, and they have knowledge that we don't, especially about reproduction, biology, behavior, and we didn't have. Most of us in our universities, we didn't have what it takes to do this kind of experiments about reproduction, for example. So even today, you find scientists using hobbyist books to reproduce, to breed some fish, because, well, they are far better than us doing this. We are indeed terrible doing this. And because the level of attention you have, and the idea of that bowl of glass and a small betta inside with some plastic plants, this is far from the reality of the hobby. They do an amazing work, and they support us much more than the university sometimes. So they are very good companions, and my life would have been totally different without the contact with hobbyists. So I owe them a lot.
That's great to hear. Thank you. And Giran and Bella, would you like to jump in on anything there as well, or shall I move on to the next question, which is also about the aquarium hobby?
Yeah, for the aquarium hobbies, for me, hobbies are not just consumers. They can be partners in conservation. And by documenting spaces, sharing collecting data, and supporting sustainable sourcing, and they contribute to science. And so my message is simple: stay curious, but also stay responsible, because every fish you keep has a story in the wild. That's all for me for the hobbies.
Yeah, for
Brilliant.
the killifish, I also wanted to add something. I mean, many people love killifish, but there are different sorts of killfishes and notobranchius. There are not too many people playing in that hobby, but that is important because some species, like for example, there is Notobranchius steinforti, which is critically endangered. It was found in 1976 and never again. Although the locality, the type locality is clear, it's precise, and many people have been at that place and trying to find the fish again, and nobody could find it again there and in the area, in the same drainage system. So that species is surviving in the hobby, by few people who are breeding it again and again since 76. So that is an important part in the hobby as well. Otherwise, we would not have an idea about what is this fish and what is this relationships and phylogenetic analysis and things like that. So I think that also for killifish, this is a very important aspect.
Brilliant, thank you. And leading on from that, I'm keen to hear your thoughts on the sort of sweeping idea on whether the aquarium trade helps or harms biodiversity discovery.
I mean, if you ask me so for killifish, the aquarium hobby is very positive in my opinion, and there are also hobbyists going into those countries where killifish live in Africa or also in South America, and they contribute a lot to the diversity, the current knowledge, and the diversity of the species. Uh, for killifish, there is never a kind of mass collection, right? So just a couple of pairs, a small group, which is taken away as live collection, and then that small group survives through generations for many, many, many years, like the example I just said before. So in terms of volume, it does not impact. And in terms of scientific insights, it does impact. So I think it's a positive aspect.
I would like to add something about the topic. I think, well, we have good and bad people in all areas you pick, So we have good people working and exporting fish, but we also have bad people doing this. I think in general, when you look to the bigger picture, you can see much more benefits than harm. I think yes, we have bad people doing this. In South America, for example, we have people fishing with poison, for example, which is very harmful. So this is something we must avoid. But working with people from Europe, from the United States, you can see they're very serious, and most of them don't want fish captured illegally. They don't want to support this. So they're always looking for the best suppliers, and also because the fish is better when you don't fish them with poison, for instance. So I think in the bigger picture, we have much more to gain from the hobby than to lose.
Great. Joanne, did you want to add to that as well?
Sorry?
Sorry, did you want to add to that as well, or shall I move on to the next question?
What question? Sorry, I can hear you.
Oh, no, that's okay. I just wondered if you had any opinions on whether you feel the aquarium trade as a whole benefits or harms biodiversity discovery.
Okay, for my case in Indonesia, as for a story as for Indonesia, if from compete while continue to describe new species, many challenges remain, especially in understanding their ecology and ensuring their long-term survival. Moving forward, conservation efforts for Stipodon require strongly collaboration between researchers, local community, policymakers, and even the aquarium trade, because only through collective effort and continued research can we truly protect this unique and fragile species for the future. Maybe from me. Thank you.
Great, thank you. So everyone's in agreement that the aquarium trade is a definite net benefit to biodiversity and biodiversity discovery. Brilliant. I know that there are some people who represent sort of the aquarium hobby on this webinar. So yeah, it's great to sort of have that alliance. Another question for Jiran. In your opinion, are there, not even in your opinion, did you know for a fact, like Luis was saying about the undescribed Corydoras species, are there more undescribed goby species that are currently in the aquarium trade?
Yeah, yes, very likely. Based on my interaction with collector and hobbies, I frequently encounter species that do not match any described taxa. This suggests that biodiversity, especially in the region like Indonesia, is still underestimated. And in fact, the aquarium threat can act as an early detection system for new species. And so many photos from the hunter and hobbies sent to me. I said, oh, I think this species is unique, it's different, but it's difficulty to find the type locality. Some hunter is make secret the type locality to reducing the competitive with other hunter. That's why it's difficult to find the type locality some of them. But I found some species. I think this not this undescribed species, goby especially, is for me.
Brilliant. And Bella, what do you think about killifish as well?
Yeah, there are still a number of undescribed, around there. Actually, I do have at home a few more Notobranchus undescribed, and also that's my first or primary interest. And then I think I also told in the new species publication that my secondary interest is for lampies, so Lacustricola, micropanchax, and so on. And I also do have a few undescribed species. I'm working on them from those. And Michael, I think you said it at the beginning or after my presentation that if a new road is opening up or a new area is opening up, then there is worth investigated, and this is exactly like that. That every new area, when we have the information that now it is possible to go there, where for years or decades it was not possible to go there. And in that case, we are almost sure that you will find some interesting things there because the area was not investigated. And that is certainly what is happening also for killifish in Africa. Now, there is a second point to that. When I said that 70 something percent of the fish are already in a threatened category in terms of conservation status, all those new species, they are not well known, they are not widely distributed, they are coming from a small area. And, by default, they will be endangered or vulnerable just by that fact that they inhabit, a relatively small area, that small area which was opening up. And therefore, we are adding more and more threatened species, to the described species.
Brilliant. So you have a few undescribed lampies and killifishes at home. Watch this space for new species 2026.
Yeah.
And Bella, you mentioned that more than 70% of Nothobranchia species are threatened with extinction. Last month in February, shoal partner David Alila at the University of Dar es Salaam, I believe a friend of yours, has finished a Rufford Foundation funded project in the Mbezi River Basin in Tanzania to assess the conservation status, habitat condition, and threats facing 7 endemic threatened Nothobranchia species there. The results are alarming, sadly, and the seasonal wetlands the killifishes rely on are being erased or transformed, which is putting immense pressure on the species. So, Bella, in your opinion, what kind of conservation initiatives need to happen to ensure these killifishes in particular, but all sorts of species, aren't lost?
Yeah, thanks for that question. So first of all, I know David, and when I was collecting several times in Tanzania, then I also invited him, and we did some collections together. So I know him also personally, and we are in contact, and also with Tim Davenport. I think they are working together in Tanzania on some projects. So general, that the biggest threat for freshwater fish in general is habitat loss or the habitat which gets deteriorated. And that is the biggest problem. And exactly like what you described, that there are wetlands, and for Notobranchius fishes, those are seasonal wetlands, which are converted into agriculture land. So when I was working in those African countries and working together with the local people, professors, researchers from local universities, then we have been thinking about, a kind of step-by-step process. And the first step would be to create awareness, and then to just simple examples, like just create some big signs which describe a little bit, the life cycle of the fish, which could be also combined, by the way, with the life cycle of mosquito. Mosquito is a big problem in Africa, malaria and so on and so on. And this fish actually eats the mosquito. So if we could create, combined signs where the two life cycles are combined, so that the fish in one stage is eating aquatic phase of the mosquito, and put it out there, that would be a very good first step. People do not want to harm those habitats or do not want to harm the fish. They do not know enough for that, to protect them. And therefore, we will need to create awareness that, they do not need to disturb, the totality of the habitats, because there are good things, living things living in the water, which are helpful for them. So against mosquitoes, for example. And therefore training in local schools and to get them know what is underwater. They know much better, the butterflies and the mammals and many other birds and many other animals which are well visible. But I observed that, of course, fishers, they know the fish, but other people not necessarily know about the fish, and fish are also important part of the ecosystem.
Yeah, so a lot of it comes down to the sort of community engagement on the sort of grassroots level. And like you say, people don't intentionally want to harm species, but a lot of it comes down to education and awareness.
Yeah, as a first step, obviously, and of course, we will need to protect key habitats and many other things, right? But we could already create something simple to start with.
Yeah, brilliant. I don't know if Jiran and Luis, you want to add anything to that or not? There is one final question from Stephen addressed to Luis. Are there plans to go back and try to find CW111 again?
Well, yes. Thank you for your question, Stephen, and thank you for your support. Yes, but this fish it's pretty difficult to get because the locality is a mystery right now. So it's a very expensive trip. So unless we have good information on the exact place, I think it's very risky to go there without this kind of information, considering the cost of the trip. So yes, maybe one of my ideas is to work with F1 specimens. This is a possibility to. So again, working with hobbyists. So I think this will prove to be easier than to collect them, because some fishermen, they don't share the information with us because they are specialists in collecting that specific fish. So they don't want to share the locality with us, and they know we provide we can provide approximate coordinates, but we will narrow the place in the publication. So they are very worried about this.
Fantastic, thank you. And a question to the floor, anyone can jump in. In your opinion, what steps should be taken to ensure sustainable use of the fishes that you talked about and fishes in general?
That's for me?
Sure, it's for you. Thank you.
Well, I think the hobby itself it's a very nice start for this. So we have today we have some hobbies breeding some species that are rare in natural habitat. So for conservation and also they do something very important, that is to make artificially rare species. Sometimes the species is rare because they are exported in small numbers, and they eventually breed them in good numbers. So the species becomes common in the hobby. So the pressure in natural habitat is very small right now for many species because they have a good stock in the hobby. So I think a good start is to work with them towards conservation because they are doing a great job breeding this species, which is something we have tried many times, and we have just a couple of examples in the academy right now having success with this. And even these examples, just few examples, they only did this because of the work in partnership with hobbies. So I think for conservation and to help the natural populations, the hobbies is playing a very important part in this.
Brilliant, thank you. And one final question again about the aquarium hobby, this one to Jiran. What message would you like to give to aquarium hobbyists? You're muted, Jiran.
Yeah, sorry. Yeah, so message for the aquarium hobbies for me. Yeah, this before I told you about the hobbies is very helpful for me to until today, except including the describe some new species and detect some species in the trade, especially. And yeah, for hobbies or and trade, they can be partnered in conservation actually. That they are documenting species, sharing and collecting data, and support sustainable searching. They contribute to science, and for them stay curious and stay responsibility. And yeah, I need to collaborate with hobbies and hunter also for my career on the freshwater. This for me.
Brilliant, thank you. And I actually have one question that I thought of for Bella. It's not an important question, but an interesting one for me. I think it was Northobranchius brayeni. You were just talking about how terrible the roads were and how challenging it was to use the car. But then there was a train station. Were you not able to, was there not a train that you could take deep into the jungle?
Yeah, look, I think it takes like a few days or a week to get from Lubumbashi to that station on the train. This is what I heard, and it is also a complicated way. Without much food or without even a toilet or something like that. So it might seem easier, but not so much.
Okay, yeah, that makes sense. It's a remote and challenging place to get to, however you decide to get there. Final question, guys. Thank you so much. Where can people find out more about your work? Is there any way you'd like to signpost people to learn more about what you do?
So if I just continue, thanks for the question. Obviously, I'm on ResearchGate. If someone is interested, but as I said in my presentation, if you go to my webpage, Wild Notos, Notos is the abbreviation of Notobranchius, so Wild Notos. Then you can find all the publications there, scientific or books or other type of publications. So thanks for that question. If anyone's interested, could find there much more information. Otherwise, I will continue to come up with some new species.
For me, you can find about my work also on ResearchGate, as Bella said, and I also have been quite active on Facebook and sharing because most of the fish groups are on Facebook. So I share my work on Facebook and also on Instagram. So you can find most of them on these social media.
Brilliant, thank you. And Jiran as well?
Okay, for me, I think it's same way to me. You can Or find me on my research get also for my doing research about Amphidramus gobies especially, or some species from Sulawesi region I did. So I'm active also on Instagram and Facebook, and if you same with the Luis, I share with the hobbies and hunter on the social media, and so we can share in on the platform. That's from me, Michael.
Brilliant. Well, thank you so much, Bella, Jiran, and Luis. It was really fascinating hearing about your talks and your adventures. So thank you so much for sharing that with us. Just to wrap up now, if anybody in the audience has any questions that weren't answered during the webinar or that we didn't have time to answer, please email them over to info@shoalconservation.org. I'll forward them on to the panelists and get back to you very soon. And if you're a freshwater fish researcher yourself, or know of anybody who is, and you know of any fishes newly described by science that you'd like included in New Species 2026, please do get in touch anytime through the year. As you know, the webinar's been recorded. Feel free to share it over social media with Shoal tagged so that we can repost. If you'd like to rewatch it or know of anybody who was hoping to watch it but couldn't make it, you'll be able to find it online from tomorrow at youtube.com/@shoal_org, or go to the Shoal website, shoalconservation.org, news and stories, and then videos. If you haven't already, please sign up for Shoal's monthly newsletter at the website. That will keep you updated on news from the wider world of freshwater species conservation, and give us a follow on LinkedIn, Facebook, and Instagram. Through April, May, and June, we'll be hosting 3 webinars as part of the season on springs, where we'll speak with experts around the world about all things freshwater springs. In April, it's the 23rd of April, I think. We'll kick off the season with a session about the importance of springs, including ecological and cultural significance. And then in May, we'll host a session on spring biodiversity and research. Following up in June will be a session on conservation and policy for springs. We'll confirm the details very soon, so keep an eye on Shoal's comms channels. It'll be fascinating. I've been learning a lot about springs recently, and they're really interesting ecosystems, so we hope to see you there. And that's it for now. Thank you so much for joining us. Thanks again to the panelists, and I hope you enjoy the rest of your day, and I hope to see you next time.
Thank you,
Yes.
guys. Thank you, Michael.
Thank you very much,
Thank you.
Michael.
Thank you.
Bye.
Bye.